All right, so now we're going to start talking about cellular respiration in more detail. Now, cellular respiration is going to be the aerobic harvesting of food. Harvesting of food energy. Okay, and what this means, this word aerobic, this means that there is oxygen. Okay, so cellular respiration, you'll want to start thinking of respiration, so that's breathing. So what we actually see is we are going to kind of take oxygen into our bodies. The oxygen is then going to travel into the bloodstream and is going to be delivered to the cells, all cells in the body. Okay, this one's just showing muscle cells, for example. In the cells themselves, we're going to have cellular respiration take place. Okay, as a byproduct, we're going to then have CO2 produced. CO2 then needs to go back into the bloodstream, back to the lungs, and we are going to breathe it out. And so when you think of respiration, we think of breathing. Well, this is the actual what's happening on a cellular level. Oxygen is being taken in. We're then using it during cellular respiration, and we'll talk about its function. And then we have this byproduct of CO2 that we need to get rid of, okay? So this is going to be kind of where that name comes from. We're going to talk about a type of process where you don't necessarily need oxygen, but most of what we're talking about today requires oxygen. Okay, oxygen is a very important molecule and is going to allow us to actually take the glucose and break it down. Now, the process of this is going to take place is we're going to do a series of things called redox reactions. Okay, and I don't want you to worry too much about this, and this is just an image of what redox means. So this is the actual chemical reaction for cellular respiration. So you have glucose, you're going to have oxygen, but you have 6 of them. You're then going to have, as a byproduct, 6 molecules of CO2 and 6 molecules of H2O. If you counted this out, there should be the same number of carbons, hydrogens, and oxygens on both sides. Okay, so you can actually do that. Now, what's happening is we're going to take glucose and we're going to oxidize it, which means we're going to actually strip the electrons and these hydrogens that are attached to the glucose off. Okay, and so when we oxidize something, we are kind of taking the electrons away from a molecule. So in this case, what's going to happen is we're going to take the hydrogens off of the glucose molecule. In that process, we're going to eventually get to a point where all of the hydrogens have been removed and all you're left with is carbon dioxide. Okay? Now, this oxygen is essential for this whole process to take place. The oxygen is going to be called the final electron acceptor. Oxygen is extremely grabby. It wants to grab onto any electrons it comes into contact with. And so what's going to happen is oxygen at the very end of this whole process is going to grab electrons in the form of hydrogens, and kind of when it joins together, they're going to make a bunch of water molecules. Okay, and so this whole process does these redox reactions, and so I can call this redox, which basically means oxidation and reduction. Okay, when something is reduced, it's gaining electrons, which means it's actually getting more negative. That's why we call it reduced. This oxygenation, what this means is that you're actually the glucose is losing electrons, okay? And you end up with kind of the oxygen left with the carbon. So this whole process is going to take place doing these redox reactions. All right. So I wanted to show you a brief overview of what cellular respiration actually looks like, and we're going to break it down and kind of go into a little bit of detail for each of these stages. The first stage is going to be called glycolysis, and this is going to take place in the cytoplasm, okay? We've talked previously about mitochondria. They are going to be where most of cellular respiration takes place. So this is going to be the mitochondria right here. But the very first step of cellular respiration takes place outside of the mitochondria. So this is going to be called glycolysis, okay? And when the process is going to what's going to happen is that we're going to have glucose. It's going to be turned into a different molecule. After the first step, we can then go into this other step, where we're mainly going to talk about the citric acid cycle, but we have to talk a little bit about this other step, which is taking the end product of glycolysis, converting it into a new molecule that then can go directly into this thing called the citric acid cycle. The last step is called the electron transport chain, which we will talk about. This is going to be the major place where we make most of our ATP. You're going to notice that down here we have ATP amounts. We make about 38 molecules of ATP during cellular respiration, but that can vary depending on many things. I'm never going to ask you what the number of actual ATP molecules is, so don't worry about that. One thing I want to mention right here is that you're going to start seeing these molecules, and these are going to be electron carriers, okay? So on the next, I'm going to start writing on here, but I want you to notice this NADH, and we have this thing called FADH2. So one thing about energy, so we've talked about energy, is it can store energy for different periods of time. So we have long-term storage, which these things are going to be like starch, glycogen. So energy can be stored in these forms for very long periods of time, okay? We also kind of have a medium term storage. We can think of glucose, okay? And then we have these kind of short-term storage molecules. And that's going to be something like ATP, okay? We also have what we call these electron carriers. And in cellular respiration, the two we're going to talk about are going to be NADH and FADH2. So what these two things are, and they usually are drawn like this in these cartoons, is they carry electrons, okay? So electrons are going to actually be stripped off and they're negative, and when they hold them, they're going to have their little electrons in these carriers, okay? So originally, NADH starts off as something called NAD+. So it has a positive charge on it. Positives attract negative, and so when there are electrons that are just floating around, they will actually bind to the NAD, okay? And then when they do, these electrons are carried in the form of hydrogens. They will then form this NADH. The same thing happens with FAD. The main thing you need to know is that they carry electrons, okay? They're short-term carriers of electrons. They'll take electrons from one place to another, and all of the electron carriers in cellular respiration are going to take electrons that have been taken off of glucose, off of acetyl-CoA, off of what happens in the citric acid cycle, and takes those electrons, these little negative charges, to the electron transport chain. Okay, and it's called the electron transport chain, so it has to deal with electrons. So NADH, FADH2, these are electron carriers. Okay, so just important when you see them. When we talk about photosynthesis, I will mention one other name that has a similar kind of, it's actually called NADPH, but it's the same function. They carry electrons. All right, so we're going to now go into the actual steps of cellular respiration. So I mentioned previously that the first step is called glycolysis, so we're going to talk about this stage and the kind of what's happening. All of these steps are actually quite complicated, but we are going to go kind of with just the most important parts that I want you to understand. All right, so glycolysis is the first step. So that's one thing, is to always know what order these go in. So glycolysis is always going to be first. This takes place in the cytoplasm, not the mitochondria. Okay, this also does not require oxygen. This is going to be important at the end. Okay, so I'm just going to say O2. All right, the input of glycolysis, so during this process, the input is going to be glucose. So this is where we're going to start using that glucose. And the output is going to be a molecule called pyruvic acid. And we're actually going to have two of them. So for one every one glucose molecule, you have 2 pyruvic acid molecules. This process of taking glucose, and we're going to start breaking glucose apart, it does require ATP. So I'm going to write ATP right here because it requires 2 ATP. However, during this whole process, what's going to happen is we're actually going to make a little bit of ATP. We also are going to have NAD plus go into this, and at the end of it, we're going to have some NADH that is going to be produced. Okay, and so these are going to hold electrons. This will eventually go to the electron transport chain, and I'm going to call it the ETC, electron transport chain. And I can spell that out for once. Electron transport chain. So that's going to go to electron transport chain. This whole process is actually going to produce 4 ATP molecules. However, we use 2 at the beginning. And so what we usually say is that, well, we have to subtract the 2 that we started with because we had to use those. So it has a net gain of 2 ATP. Okay, so this is going to be what happens in glycolysis, and I'm going to show you what this actually looks like. Okay, so here we have glucose. Glucose is a 6-carbon sugar. It's going to go into this process called glycolysis, so this whole thing is going to be called glycolysis, and these images will be on the PowerPoint that I'm posting. It requires ATP. The first step is that we actually split the glucose molecule into 2 3-carbon structures. We are then going to remove some of the electrons from these. And you don't really need to understand how that happens. Just know that electrons are removed. They're going to go to NADH. Remember, so we have 2 of these happening. And then we're going to be able to produce a little bit of ATP at the end. The final product is this thing called pyruvic acid. So your main goal to understand this is that you start with glucose, you end with pyruvic acid. It takes place in the cytoplasm, okay, and it does not require oxygen. Okay, so again, cytoplasm is where it's going to take, cytoplasm, where it takes place, and it does not require energy, or sorry, does not require oxygen. All right, so from the end of glycolysis, we're going to now go into the next step, which is called the citric acid cycle. So here's the citric acid cycle, and what you're going to see is that there's this little step in between. So we have pyruvic acid that's produced here. The pyruvic acid is actually going to go into the mitochondria, so the rest of this is all going to take place in the mitochondria. Prior to being able to do the citric acid cycle, we have to change pyruvic acid into a different molecule. Okay, so let's kind of go on to our next step. So we'll say citric acid cycle, citric acid cycle. Okay, this is now going to be in the mitochondria. Okay, and so pyruvic acid that we produce at the end of glycolysis is going to be converted into another molecule. And so this is a lot of steps. There's lots of enzymes involved in this. But it turns into something called acetyl-CoA. Okay, so remember this one. This molecule can go directly into the citric acid cycle. Okay, so we'll see what this little process looks like. So we start off with a pyruvic acid, and remember there are 2 of these, so this will happen twice. Pyruvic acid actually loses the carbon dioxide and it becomes this molecule called acetic acid right here. So acetic acid is 2 carbons, and we're going to have this thing called coenzyme A that comes in. It actually binds to the acetic acid. During this process, we actually have NAD plus that is going to take some of those electrons, because again, we have the CO2 that has been given off. And at the end of this whole process, we have this thing called acetyl-CoA that's produced. This is the only thing that can go into citric acid cycle. Pyruvic acid cannot go in by itself. Acetic acid can also not go in by itself. It can only go into the process of the next step through acetyl-CoA. All right, so once we are actually into the citric acid cycle, the input, okay, so this is going to be the input into citric acid is going to be the acetyl-CoA. And the output of this whole process is going to be, we're going to have a waste product of carbon dioxide. So this is where we basically have broken glucose completely down into carbon dioxide. We're also going to make some ATP from this process, not very much, but a little bit. And we're also going to have a bunch of electrons that are going to be transferred to the 2 electron carriers that we talked about. So we have FADH2 and NADH. So this is going to be what then can go into the electron transport chain, which is our last stage. So this is going to go to last stage. The CO2 needs to be gotten rid of, which is going to go back into the bloodstream and actually we'll breathe it out. So this is what the citric acid cycle looks like. And again, these are very complicated. We're just going to go very basic. Okay, so we have this acetic acid, which is eventually, it's turned into acetyl-CoA before it can go into this. But then once it's in here, it's going to turn into something called citric acid, which is why we call it the citric acid cycle. And there are multiple different steps that we go around. But basically what's happening is you're breaking apart the molecule, attaching it to different things. But the main thing that I want you to know is the output, okay? And you don't actually need to know kind of the numbers of each of these, but just know that you have carbon dioxide, which is a byproduct. We have ATP, and then we're gonna have these 2 energy electron carriers, okay? So NADH and FADH2, okay? Now, one thing I want to just mention is, we keep on breaking down glucose, and what's happening is that we're kind of taking the electrons that we're taking them from the glucose molecule, and as they fall, they're going to release energy. And so I wanted to mention this briefly, this idea. Okay, so I'm just going to write this out. During cellular respiration, electrons gathered from food so basically glucose, some molecules gradually fall, losing energy at each step. Okay, and so what happens is we start with kind of these electrons that are attached to glucose. They then move to something called, the NADH. Okay, these are now going to go to our next step, which is going to be the electron transport chain. Okay, and then finally, the electrons are going to fall all the way to oxygen. Okay, and if you remember, I said oxygen is this is going to be called the final electron acceptor. It is what we call an electron, oops, that is not right, electron grabber. It really is attracted to electrons. It wants to fill up, if you remember, oxygen has those 2 extra spots in its outer shell, and it wants to fill up those with electrons. Okay, now you have to be careful with this because you can't just go from glucose to oxygen directly, because that would be the energy is lost that way. And so what happens is instead you take the electrons removed from glucose and you step them down multiple steps. They go through these redox reactions where you're kind of taking them off of one molecule and putting them on another. And so they're going to continue to fall. At each step that they fall, they release ATP, okay? And the final place that they're going to end up and they'll be kind of attracted to is going to be the actual oxygen. Okay, and so when oxygen comes into contact with these electrons, it's going to form H2O. Okay, and so this is kind of a very interesting process, and it's kind of hard to understand. But here we see it in the actual process. So electrons from the food, they are going to be added to NADH. Okay, the electrons are now going to move to the next step, which we're going to talk about, which is the electron transport chain. But we're going to actually step them down from molecule to molecule. And as we move these electrons, they're going to produce ATP. The final step right here is going to be when the hydrogens, electrons, and the oxygen combine to form water. All right. So this is what the electron transport chain actually looks like. And it's probably you're gonna say this looks really complicated. And it's pretty complicated, but what the electron transport chain is, it has these proteins that are built into the membrane. Okay, and so we're in the mitochondria still, and I'll write this out for you guys, but we're in the mitochondria, and the mitochondria has kind of this double membrane where there's a space in between the membranes, and then there's kind of this other outer space. Okay, the proteins are going to be built within the membrane. So we have these molecules, NADH, that have electrons. We have FADH2 that have electrons. And what the electrons are going to do is they're actually going to take the electrons from the NADH and give them to these proteins on what we're going to call this electron transport chain. When this happens, it provides energy to pump hydrogen ions, these H pluses, from one side of a membrane to the other. Okay, this is going against their concentration gradient. So they're having to go against, there's less of them here. They're trying to push them out into this area where there's more of them. This requires a little bit of energy, and the energy that is allowing for this to happen is the movement of those electrons moving down, and when they fall from one space up to one lower, they will give off energy. So we're pumping hydrogen ions out into this space between the membranes. Okay, and then we have this really high concentration of hydrogen ions that forms up here. Then what happens is we have this molecule over here called ATP synthase. ATP synthase acts like a turbine. Okay, so if you've ever seen how dams work with water, you have water that's kind of built up, and I can try to draw this probably not very well, but you have a dam, so here's our dam, and then water is back here, okay? And if you open these little holes, water can start to flow through. As they fall out, they can sometimes turn these things called turbines, and as they are spinning, they will produce electricity, okay? And so what happens is we have this protein called ATP synthase. It's actually an enzyme that is going to allow for hydrogen ions to run through it, and it kind of spins, and as it has these rush back through, they produce ATP at really high quantities, okay? And so this is what the electron transport chain looks like. So let's write a little bit of that down. Okay, so we have electron transport chain. Okay, so there's proteins that are built into the inner membrane, of the mitochondria. Okay, the energy stored by the electron transport chain behaves like the water behind a dam, okay? And then the turbine structures are going to be the ATP synthase. Okay, so again, what's happening is we have these electron carriers. They go to this thing called the electron transport chain. Electrons are going to be donated to these electron carriers. As they fall, they're going to allow for energy to pump hydrogen ions against their concentration gradient from an area of low concentration to an area of high concentration. They're going to continue to move from one of these molecules to the next, okay? As they do that, pumping more and more hydrogen ions through. The hydrogen ions are then at a very high concentration in this inner membrane space, and then are going to be able to run back through this special enzyme called ATP synthase. Think about this, ATP synthase, like synthesizing, it synthesizes ATP. As the hydrogen ions run through this, it turns the ATP synthase, creating ATP. So this whole process is happening. The only reason why this will work is because we have this really high concentration gradient of the hydrogen ions up here. Okay, so they will naturally run through this way because this is where there's less hydrogen ions. Okay, so this is the last step of the electron transfer chain in cellular respiration. Down here, what you're going to notice is that the electron carriers, the electrons move from each of these down, they're kind of down from one to another. The final electron acceptor is oxygen. When oxygen comes into contact with hydrogen ions, it then produces H2O. The oxygen is the final electron acceptor. Okay, so here's the electrons, here's the final electron acceptor, which is oxygen. You must know that, okay? Now you may be wondering why there is this skull and crossbones right here. So what happens is this is the location that cyanide acts as when it's doing its damage. So cyanide is very dangerous and it is a poison that will kill you. So what happens is if you ingest cyanide, it attaches to the electron transport chain in this location right here, preventing this from happening. Okay, and so it prevents the electron transport chain from working, which means that you can then not make any ATP. If you're not making ATP, you will not stay alive for very long. Okay, so this is going to be kind of the end stage and something you would not want to mess with in this case. All right, so that's kind of the overview of cellular respiration. Okay, so I want you to just remember, so cellular respiration. Okay, so we start with glycolysis. We put in glucose. We make pyruvic acid. Okay, we also are going to produce a little bit of NADH because we have some electrons that are taken out and a little bit of ATP. We then are going to go to the citric acid cycle. Okay, pyruvic acid first has to be turned into acetic acid, and then we have something called acetyl. Oops. I forgot how to spell that. Acetyl-CoA, and then the acetyl-CoA will go through the citric acid cycle, okay? And at the end of this, we're gonna have basically the byproduct, so acetyl-CoA. I guess that's how I spelled it wrong. Oops, that is really spelled wrong right there. Sorry. Acetyl-CoA will then go into the citric acid cycle, and out will come carbon dioxide. We'll have NADH that's made, FAD, FADH2, and ATP. And then this stuff goes into the electron transport chain, okay? So 3 electron transport chain. Okay, so this is where we make a bunch of ATP, and we also will make water at the end, okay? Now, one thing I wanted to mention is that when we're talking about the final steps, so both the citric acid cycle and the electron transport chain, those require oxygen. And so we have situations where if there is no oxygen, we're gonna do something called anaerobic respiration. Okay, and what this means is that there is no oxygen. Okay, and in front of aerobic, so this means oxygen, means no oxygen. Okay, so this is the process of harvesting food without oxygen. Okay, so what this process does is all it does is glycolysis. Okay, and the process is called fermentation, which you may have heard before because it has to do with how we make a lot of our food and beverages. So glycolysis does not require oxygen, and it does produce 2 ATP. Okay, so this can be used in situations where there isn't enough oxygen, and it allows for a little bit of ATP to be produced. Okay, now one of the major issues with kind of glycolysis is that the end product, usually we're making pyruvic acid. So under normal conditions with oxygen, in glycolysis you make pyruvic acid. However, one of the issues is when there's no oxygen, you have a problem with your NAD+. Okay, so in aerobic respiration, NAD+ is recycled. Okay, so normally what happens is NAD accepts electrons and then turns into NADH. But this NADH has to go to the electron transport chain before it is going to be able to turn back into NAD+. Okay, because it has to drop off its electrons in the hydrogens. So what happens is when there is no air, this is not going to be able to recycle in the normal way. And so instead of giving its electrons to the electron transport chain, it adds them to pyruvic acid. So I'll say this right now. So under anaerobic conditions, NADH donates electrons to pyruvic acid. Okay, and so when that happens, pyruvic acid is going to turn into something called lactic acid. And this is what happens in humans. So you probably have heard of this before. This happens in our muscles. So if we're working out really hard, we're not quite getting enough oxygen to our muscles. Our muscles can start to do glycolysis, and if there's not enough oxygen to complete the entire process of cellular respiration, the pyruvic acid will have electrons donated to it by NADH, okay? And so what this looks like is here. So we have our glucose. So this would be like we have this, the glucose is there. We do not have oxygen, so we have glycolysis that takes place, okay? We have a little NAD+, the NADH is formed. The NADH has nowhere to go because it can't go to electron transport chain if there's no oxygen. And so it's just sticking around, and there's a bunch of pyruvic acid around. And so what it does is it instead takes its electrons, adds them to pyruvic acid, and turns them into lactic acid. Okay, this lactic acid can then kind of feel like a burn in your muscles. And, at some point, you can't continue to do this. You actually have to have oxygen. So if too much lactic acid will build up, you'll have muscle fatigue, and at some point you won't be able to kind of move on too much with that. All right, now this process of fermentation takes place in lots of different organisms, not just in us. And so fermentation is gonna be always this process of kind of glycolysis, in the absence of oxygen, okay? So when other organisms like yeast or bacteria do fermentation or this glycolysis, they don't always make lactic acid. Some of them will actually produce something called ethyl alcohol. And this should sound, we may not have heard it called this, but this is the alcohol that we actually will drink. Okay, so beer, wine, all of these are going to be produced in this process of fermentation where you take these microorganisms, you're basically forcing them to do this fermentation in the absence of oxygen, which then have these byproducts. So things like soy sauce are produced this way, cheeses can be produced this way, yogurt, all these things through this process of fermentation. Now, the last thing I wanted to talk about is that lots of microorganisms do this, and we often will utilize their byproducts, but we have to think about what did the earth look like originally, okay? And we'll eventually talk about how photosynthesis is extremely important for life on earth because it produced oxygen. But for a long period of time in the world, there actually wasn't very much oxygen present. And so some of the very earliest organisms are going to be functioning in very little oxygen at all. Okay, so it wasn't until about close to 2.5 billion years ago that the first atmospheric oxygen is going to be present, which means that the oldest prokaryotic fossils are most likely doing things like glycolysis. So glycolysis, that one process, is a universal energy producing mechanism. And if you think about it, for us, when you do this process of glycolysis, 2 molecules of ATP is not enough to keep us functioning. But if you think about something that's a microscopic organism, 2 ATP would be completely fine for them to then be able to survive on. Okay, and so the idea is that, it's not until we get to a point where there's lots and lots of oxygen on the planet that there can start to be this aerobic harvesting of the actual glucose, okay? So this whole process is, cellular respiration. I want you mainly to know the major things, what are the inputs, what are the outputs, and there'll be the study guide that will explain those things, okay? Thank you very much.